right, hi everyone. I'm Fritz, a research scientist from OZ Technologies, and today I want to talk to you about data transformations for effective visualization of single-cell embeddings. A quick disclaimer before I start: although several authors, including myself, are from OZ, the work has been mainly done at Fred Hutch, is published in a peer-reviewed journal, and can be accessed freely and openly on the web. So with that out of the way, in a nutshell, the data transformation that we developed enables us to create embeddings that provide a higher resolution view of computationally derived clusters that in our case represent cellular phenotypes. And so here, for instance, what you can see on the left side is that with a standard embeddings, the colors in this case represent phenotypes, and they kind of blend together, while on the right side you see that the clusters are way more distinct thanks to our data transformation approach. But what are single-cell embeddings? So single-cell embeddings are lower dimensional representations of high-dimensional single-cell data. And so think of having a table like that, where each row represents a cell, and the columns are cellular features from the proteome, genome, or epigenome of the cells. And now the embedding is a representation in 2 dimensions that tries to capture the similarity of this high-dimensional data. And people often visualize these embeddings in order to get an overview of the entire data set and see how cells relate to each other in order to generate hypotheses, annotate clusters, or compare different cell types. And now there are different methods to create these embeddings. And here are 3 real-world examples showing, for instance, how surface marker expression, gene expression, or accessibility peaks can be used to construct these single-cell embeddings. And while these visualizations provide great opportunities for exploration, there are still challenges revolving around like specific or rare cell populations, as well as batch effects. And so, for instance, are we interested in just looking at broad patterns that tell us or reveal general cell phenotypes, or do we want to be able to visually differentiate more specific cell phenotypes found by our clustering approach? Also, when embedding multiple samples, how shall we actually handle batch effects? So are we interested in sample differences, or are we interested in cellular differences? And more generally, an overarching question is, are we using these embedding visualizations to explore or explain the clustering results? And this can be challenging to achieve both, because there can be a disconnect between the data representation that was used for clustering and the data representation that is used for actually embedding these data. And so to address this, we have developed a method that annotates the raw expression levels. And without going into technical details here about our FAUST clustering approach, the high-level idea is the following. So for each cellular phenotype, like these surface markers here, our method annotates the expression levels with human interpretable labels, like positive or negative. For instance, in this case of these bimodal expressions here. And doing this allows us to actually easily group cells together in meaningful and also interpretable clusters. And if you're curious, I've linked the paper at the end of my presentation. But for the visualization approach here, what's important is that now every cell of our data set is fully annotated. And so what we can see here now is that if we again visualize, the color here visualizes the cell phenotypes, we see that there's a lot more information in our data set that is not being represented currently in the structure of this embedding. And so the question is, how can we reveal the detected phenotypes, not just by color, but also structurally? And so that brings us to our annotation transformation approach, which is actually fairly simple. It consists of 3 steps. So first we remove outliers, then we normalize the expression ranges, and finally we align those expression ranges. And to give you a concrete example, let's say we look at T helper cells, which are CD3 and CD4 positive and CD8 negative. So the first step that we do is we just simply linearize the data in order to remove outliers. Then we normalize the expression ranges to have 0 mean and unit variance. And then finally, we align those expression ranges again based on the annotation level that we got from our clustering approach. And if we transform the data in such a manner before embedding it, we are able to actually reveal the clustering information structurally and not just by a color, as we can see here on the right side. And in this case, we used UMAP to create the embedding. But we can achieve a very similar effect, not just with UMAP, but with other methods too. So here we use t-SNE together with our transformation approach, and we virtually get the same results. And we can even use something else like a variational autoencoder, which on its own doesn't show too much and is probably overkill. But the whole point here is to say, like, the structural information that we're able to reveal with our transformation method is thanks to that method and not thanks to like tuning or picking a specific embedding method. Beyond just revealing the clustering structure, we can also, or one other benefit is cluster coherence of our approach. So here, for instance, I'm highlighting two cell phenotypes, and these two phenotypes only differ in their CD38 expression. And you can see that in the untransformed embedding approach, they strongly overlap. Now, if we would like visualize actually the CD38 expression, we could kind of identify a trend in the untransformed embedding, but it's hard to perceive these two phenotypes as distinct clusters, while they are very distinct in our transformation approach on the right side. And interestingly, the CD38 expression has been shown to define pre-activated tumor-infiltrated CD8 positive T cells. And so biologically speaking, it could be actually useful to perceive these two cell phenotypes as distinct clusters and not as one. For joint embeddings of multiple samples, our transformation approach almost entirely removes any kinds of batch effects. And again, it kind of makes it easier to perceive cells that are of the same phenotype as unique clusters, as you can see here, while they kind of spread out all over the place in an untransformed embedding. And such coherence, again, can be useful for inspecting rare cell types that otherwise might just spread out and are not perceivable as a unique cluster. And so with that, I want to conclude with kind of like three very overarching or high-level takeaways. And the first thing, which I hope you remember from my talk, is if you create an embedding, first do the data work and think how you can represent your data before tuning any kind of embedding methods, hyperparameter or something like that. Second, use a data transformation that is closest to your objective. So if you want to better explain or understand the results of your clustering approach, then maybe our transformation approach helps because it reveals the clustering information better than an untransformed embedding. But that doesn't mean that you should use it for all kinds of exploratory tasks. And lastly, I just want to highlight, even though our transformation approach works well together with the clustering method that I quickly hinted at, but they're not strictly coupled together. So you can use your own methods to annotate the expression levels. Yeah, and with that, I'd like to thank you. We also have a poster up if you want to learn more about the approach. And there's a related talk in 20 minutes by my colleague if you want to learn more about the clustering methods, actually. Thank you.
Any questions from the audience? Please step up to the microphone.
I wonder if you could explain the align step in your transformation a little better. I didn't really get it. The align step in your data transformation, how does that work?
The outline?
Align. Like you standardize your data, but then you do something else, and I didn't quite understand how this works.
Oh yeah, so there are basically three steps visualized here on the right side. So first we just remove outliers, simply because they exist quite commonly in flow cytometry data that we use or that we work with most often. Then we normalize the expression ranges, but after normalization, you wouldn't be able to distinguish anymore between like positive and negative expression. So the critical last step is just to align the expression ranges based on their annotation. So what that means here, so we know CD3 is positive and CD4 is also positive. That already came out of our clustering approach. So after normalization, they would all have like 0 mean. So if we would embed that, that wouldn't work at all because the embedding method wouldn't be able to distinguish between the positive and negative expression anymore. So the final critical step is to basically just choose a, and it's up to you, you choose a specific location for the new mean of positive expressions and negative expressions. And by doing so, you remove kind of like the sample-to-sample differences and allow it to basically get back the differences between the positive and negative expressions.
Like in advance, you have divided your samples into classes, and you computed differential expression between the two classes.
Yeah, this is this step which I didn't really have time to go into, which is coming from our FAUST clustering approach, which does exactly that. So the first thing we do is we essentially discretize the data into interpretable levels. So instead of working with the raw expression data, we just look at like what is positive and what is negative. And then, so that's a critical first step before doing the transformation.
I see. So for some genes, you would have these bimodal distributions.
Correct.
But presumably not all.
Not for all. You can, but if you have trimodal distributions, it also works. And there's one extra thing which I also didn't have time to go into. What you can do on top of that is some distributions, you have a hard time distinguishing what their modality is. And you can factor that into the embedding method as well. So if you have like not very distinguishable expression levels, then you can basically downweight the importance of that feature for your embedding if you want to. But it was a little bit too much. We can talk about offline about that.
Okay, but thanks. Yeah, that helps.
Thank you.